Now let's talk about, now that we got done here, and I think we unmute everybody. If you guys have any questions, you guys can go, text me. You can also ask, all right? It keeps going on mute, seems like it. I don't know if you guys are muting it, but that's all right either ways. You guys can always unmute yourselves. So if you have a question, you guys feel free to ask. Or if you don't want to ask a question like that, you can also write it, right? There's a chat kind of box right there for you to use as well. Let's start talking about not our teeth, but mastication and the process of digestion. So the first step is to take that big chunk of food and then break it up into smaller bits, right? So that's mastication. Mastication, chewing with the incisors and the canines, allowing us to bite or cut off food. The molar type teeth grind the food itself, and then it's powered by the masseter, temporalis, and then the pterygoids. Remember, the pterygoids' main purpose is to kind of move your teeth from side to side, as well as to depress the mandible right here, right? So it does cause what's called a mastication reflex. The reason why we have this reflex is it kind of makes sense. What would happen if you have to think about every second and every contraction as you're chewing a bit of steak, right? It will get basically, you'll get bored to death just chewing. So we have this reflex that provides a little bit of control when we need to, when we decide when we've chewed enough, then we swallow. But before then, you just keep chewing and chewing. Think about, again, all right, if you're chewing in a hard piece of steak, right, how long do you chew before you decide when to swallow? And when you're chewing after the first bite, did you really have to think about chewing afterwards? Usually you don't, because its reflex pathway takes over. Now, besides the mastication, we have salivary glands, the parotid, submandibular, sublingual. The parotid glands are external to the masseter. The parotid glands are connected to the parotid duct, which pierces through the buccinator muscle. As it pierces through the buccinator muscle, then we can introduce that glandular secretion that is enzyme rich. So when you hear the words serous, serous means liquidy, meaning enzyme rich. It's opposed to mucus. Mucus means it's enzyme poor and real thick. So serous secretions are always going to be better when we're eating. It's also the reason why, if you've ever seen a dog that's eating or that's real hungry, or God forbid, some kids when they're real hungry, right? Sometimes when they're real hungry and they're eating, a little bit of drool comes out. Well, the drool more easily comes out of the mouth because, right, when they're eating, there's that serous secretion that's more watery. The thicker secretion, the mucus secretion, usually is too thick to just leak out. So the parotid gland is the largest in our main serous gland, meaning that when it activates, it produces a lot of enzyme-rich fluid. The enzymes takes a lot of energy to make, so you don't want to always make the enzyme-rich type of fluid. The submandibular glands are a combination. It primarily makes serous fluids, but can make mucus. So we'll talk about when we have mucus and when we have serous in a minute. Sublingual glands underneath the tongue. They have ducts that go right on the tongue, under the tongue. They're mixed, but primarily mucus. Now, mucus secretions are good because you don't want the mouth to be dry, right? We don't ever want our mouth and the oral cavity. We don't want our throat to be dry. The drier those regions are, the more likely it's going to split, and you're going to have nicks and cuts as a result. Some of you guys might have heard of mumps before. Mumps are part of our MMR vaccine. Mumps, measles, and rubella, right? Mumps used to affect a lot of kids. Measles used to affect, they said like 90% of kids that were pre-vaccination. 90% of 5, 6-year-olds were going to have measles. That's how transmissible measles is. Well, mumps, it's even worse because it causes a lot more pain, right? Next to the parotid, we're going to see, right, some branches of our trigeminal gland, our trigeminal nerve. That nerve is going to be our main sensory nerve. So when mumps, the parotid gland enlarges and gets really big and starts pinching down on some of the branches of our trigeminal, and it causes a lot of pain as a result of it. By the way, that's why you ever seen like old black and white pictures, right? They always have that picture of a little boy or girl, and they have a bonnet, right? Like a handkerchief that they have come kind of around the chin right here, and then they tie it up around the top of their head. The purpose of that is to elevate the parotid and the submandibular glands so then it doesn't constrict on the trigeminal nerve. So then it can ease the pain a little bit from getting mumps. Right? So compositions of saliva, wet food, it wets the food, so then it's easier to swallow. All right. The saliva also dissolves food for better tasting. The bicarbonate ions found in saliva buffer acidic foods. There's the big ones right here. The main enzyme that we see in saliva is salivary amylase that begins starch digestion in the mouth. Now, remember I said that we make a lot of secretions a day? Think about this. We make 1 to 1.5 quarts per day of secretions. Now, just from saliva, 1 to 1.5 quarts. If you guys have been to Leon's, if you've been to Cops, it's not the small pint container, it's the big quart containers. And you're making 1 to 1.5 quarts of just saliva a day. So kind of crazy. Now let's talk about which type of saliva we make when. There's a big one. And again, right, keep in mind our body hates to waste energy. So when we are releasing an enzyme, the enzyme takes a lot of energy to make. Would you want to release that enzyme when you are not eating? Probably not, right? There's no payoff then. So what happens is when we're eating, we have more parasympathetic input, and it causes then more enzyme-rich saliva to be produced. So you have more parasympathetic when you're eating. The parasympathetic nervous secretion activation causes more of your parotid glands to activate, and then you have more enzyme-rich solution that goes into your mouth because you're eating, and you want those enzymes to break down food particles in your mouth. Sympathetic stimulation, when you're not eating, will produce mucus-rich saliva. So what happens is when you're not eating, there's more sympathetic, right? There's always that balance between parasympathetic and sympathetic. When you're eating, more para. When you're kind of exercising, more sympathetic. And what happens when you're under sympathetic stimulation is that you don't want to waste those enzymes. So instead of producing this enzyme-rich enzyme-rich saliva, you're going to produce mucousy saliva. By the way, that's also the reason why, if you've ever seen somebody jog, right, outside, sometimes or even riding their bike, you see them spitting a lot. God forbid if you've ever watched a football player, right? Football players are gross to begin with. It's okay, I used to be a football coach and a football player. So you would see football players in the middle of a game, and I remember seeing this from a friend, right? They're playing, more sympathetic output, and then they just kept spitting. And sometimes they spit, and with football players, they have helmets that have like little, right, stuff on it, right? Little tubes around it. And it gets stuck in the tubes and it just hang there, kind of gross, right? And why? Is because, right, instead of this watery saliva that just flows right out, is this thicker, more mucus-filled saliva that's produced when you have sympathetic stimulation. Now, the rest, once we've kind of chewed it by mastication, some of the enzymes have worked its way through the saliva, and now we've broken some of the nutrients apart specifically, right, the starches, then we're going to swallow, right? Again, only starch digestion occurs in the mouth, nothing else, right? Now we're going to push it to the back of our throat, and then we're going to swallow. Series of pharyngeal constrictors allows us to push that bolus of food down into the esophagus. In the esophagus, a third of the way down there, we're going to have skeletal muscles, right? Then from that point on, it's going to be smooth muscle. The smooth muscle then's going to undergo constriction and start using peristalsis to move that bolus of food into the stomach. The mucosa of the esophagus is stratified squamish. Again, since it's connected to the mouth, we need protection. So in the pharynx, oropharynx, laryngopharynx, stratified squamish cells. In the esophagus, stratified squamish cells. And here is the stratified squamish cells right here. And these are the stratified squamish cells that don't have, right, that are, the cells are still functionally alive, right? It's there for protection. It doesn't have as much, right, kind of barriers here. It's non-keratinized, so you don't have the barrier of keratin. And its main purpose is to allow for a little bit of suppleness. At the same time, you have a little bit of protection. Next, we're going to talk about in terms of chewing. There are 3 phases: voluntary, pharyngeal, and then esophageal. In terms of voluntary phase, you decide when you've chewed enough and now it's time to swallow, right? It's not a reflex. You decide when you've had enough to, chewing, and then when to start the swallowing process. But once you kind of force that food down into the pharynx, a series of reflexes allows you to contract the pharyngeal constrictors, elevate the elevate the soft palate, so then the food doesn't go into your nose, and then move food from your pharynx into the esophagus. In the process, the food pushes down the epiglottis, which stops the food matter from going into the trachea. So here's that picture right here of the swallowing reflexes. So you push that food down into the back of your throat. That is a voluntary process. Once food hits the back of your throat, what happens? Well, what happens is food hits the back of your throat, it closes your soft palate. As it closes the soft palate right here, food then doesn't go into your nostrils and into your nasal cavity. Now, we've all had it where drink can go up this way. So if you're drinking a soda or a beer and somebody makes you laugh, right? What happens? Well, somebody makes you laugh, you have a lot of pressure from below, and it forces that fluid up into your nose. And it gives you that weird sensation, right? Where it gives you that weird sensation where you have now fluid going into your nostrils and out. Well, food is usually too large and it won't go upwards. And the reason why is because as food gets pushed into the back of your throat, it closes that gap between your uvula and your laryngo and your nasopharynx right here. So then food can't move up. Once it goes in that area, then it becomes a pharyngeal reflex, and all the constrictor muscles allow you to move that food from your throat down the esophagus. Again, it's a reflex, but it's still skeletal muscle. So if for some reason or another, you have that last morsel of taste and it's a little bit more sour than you think, right? Again, potato salad in summer, right? Hopefully we can still have potato salad in the summer. We can still have picnics this summer. But in the old days, you can have that potato salad and you decided to eat it, and then you decided at the last minute, maybe it's not such a good idea. Because it is skeletal muscle, you can still make it move up, right? But because we don't want to have to think about all the constrictions, all of the contractions, so then we swallow. Well, it becomes more of a reflex behavior. We start the process, and then our brain takes over, and reflexively contracts the muscles, allowing us to move our food from our throat to the esophagus, and from the esophagus into the stomach, right? Just because it's a reflex does not mean you have no control. You can still offset the reflex and overcome that reflex. So then if you think that food is not good anymore, or God forbid, if you have a peanut allergy, right? And you just tasted the food. At first, you don't think it might have peanuts, but the last thing you want to do is not be sure, right? You can spit it back out. So you can still have regurgitation, even though it's a reflex. You can overcome that reflex. Now let's talk about the stomach, and then start a little bit about the intestines, and then we will stop. In terms of the stomach has 3 layers of smooth muscle, very, very different than the rest of the GI tract. Where the esophagus meets the stomach, right? The area where it meets the stomach is called the cardiac region of the stomach. There are kind of constrictions of smooth muscle, right? Constriction of smooth muscles where the esophagus meets the stomach. Those constriction of smooth muscle is called the lower esophageal sphincter. This is not a true sphincter, right? This is a true sphincter. This is, all right, called the pyloric sphincter. And in the pyloric sphincter, you can pinch off, completely pinch off one region from the other region. Here, there's no complete pinching, right? It's a pseudosphincter. So there is some smooth muscle right here coming from the stomach, and it narrows that opening between the esophagus and the stomach. That narrowing makes it harder for food to go in reverse. True sphincters will not allow material to go in reverse, right? This sometimes does. You guys have all eaten something, and then a couple, hours or a couple minutes later, right, it's in your stomach already, and then it just gives you an upset stomach and you vomit it out, right? That is movement of fluid and nutrients and food in reverse, that vomiting, right? So this lower esophageal sphincter is just a thickening of the smooth muscle that prevents usually unwanted movement in reverse. But since it's not a true sphincter, when you really need to vomit, you can, right? The inside lining of the stomach right here, inner lining, right? Or the smaller lining, right? This is called the lesser curvature. The lesser curvature is connected to the lesser omentum, which is connected to the liver. So then the liver and the stomach are connected together. The larger outline of the stomach is called the greater curvature. The greater curvature is where we have the greater omentum, right? It starts at the greater curvature and it covers the rest of your small intestines. Again, cardiac region is a region of the stomach where the esophagus empties into. The difference between the cardiac region and the lower esophageal sphincter is this. The lower esophageal sphincter is a constriction, whereas the cardiac region is a region of stomach where the esophagus enters, right? So one is the kind of tightening of the smooth muscle. One is a region where it just kind of enters completely through. So the lower esophageal sphincter is found within the cardiac region of the stomach. It contains the lower esophageal sphincter. Now, other cool things, it's got 3 layers of smooth muscle, right? Now, you guys have heard of animals, sorry, animals that don't have teeth and they swallow their food whole. Think of geese. Think of, right, a snake, right, or some kind of lizard. Lizards and snakes, they swallow their food whole. And some birds do that as well, right? Pelicans do this. They take a fish, they swallow it whole, and it goes right to their stomach. Now, you might have also heard that in those animals, they have found rocks in their stomach. Now, why would they find rocks in the stomach? Simple. the rocks have sharp edges, right? Since they're not biting and they're not chewing, what they're gonna do is they have much larger stomachs with much stronger stomach muscles. So when the stomach muscles contracts, it causes the rocks that they swallowed and all the sharp edges of those rocks. It causes the rocks to start crushing that whole food matter that they've just swallowed down. As a combination of the rocks and the stomach muscles work together, that combination works very similar to our teeth in that it allows them to grind down and separate that whole animal they've just eaten. So then instead of having a whole animal now, they have smaller chunks of that animal. I know, sounds gross, right? But that's why we see rocks when we open up the stomach of large birds and large lizards and snakes. The main purpose is that the rocks in the stomach muscles, they act like teeth then, and it allows them to break apart that larger animal they've just swallowed down whole. Now, in terms of the stomach, 3 layers of smooth muscle: outer longitudinal, middle circular, inner oblique. So instead of an inner circular and an outer longitudinal, we have an inner oblique layer, and it allows us to mix and churn in 3 different areas. The rugae are folds in the stomach and seen when it's empty, right here. So when it's empty, the stomach is very small, it shrinks up. And then when you start eating, this is why it's good to have smooth muscle here, right? As you start eating, you push down the folds, allowing the smooth muscles and the mucosa to relax and accept more food. And then you can have, I mean, you guys have probably seen the people that are in that Nathan's 4th of July hot dog eating contest. They eat like 50 hot dogs in 10 minutes, which is crazy. Sometimes 60 hot dogs with bun in 10 minutes. So when they're eating that much, the stomach stretches a tremendous amount. They did a CT of somebody right after they've eaten that much food, and they found that the stomach almost stretched from one side of the abdomen to the other side. It became almost bigger than the liver, which is crazy. It shouldn't happen, but somehow they're able to get that to happen. So the rugae then will be more noticeable in an empty stomach. In a full stomach, the rugae should be almost flattened to accept more gastric filling. Now, important things to know, right, are what is found in these little openings. Now, if we look in the rugae, if we look in the rugae right here, really close, if we take a microscope into the rugae, we see little openings, right, little holes that are found in the rugae. Those holes right here are called the gastric pits. And in the gastric pits right here, again, we're looking at the rugae. If we're looking at the rugae with a real close eye and a microscope, we see that there's little tiny holes that are found on the rugae itself. Those holes are openings. They're openings that lead into what we call the gastric pits. The gastric pits are where we're going to see the cells that make the hydrochloric acid and the enzymes that we will see in the stomach. In other words, the cells that make the real caustic, strong materials, they're not found at the very top. They're found way deep inside the mucosa. We're going to see why in just a second. Right? What we do see on the surface, closer to the opening, closer to rugae, are surface mucus forming cells. So lots of mucus on the surface. And we're going to see that mucus is going to be incredibly protective. All right, so here's a gastric pit. And what kind of cells do we see? Lots of mucus forming cells at the very top, close to the opening. The deeper we go, then we see the functional cells, parietal cells, chief cells, endocrine cells. Now the parietal cells, and probably the easiest way to remember it, and it's kind of cheesy, is this. Parietal cells end with L, like that. And they produce hydrochloric acid. So I would ask about what are the cell types and what do they produce? So let's go back. Parietal cells produces hydrochloric acid and something called intrinsic factor, which is going to be important. We'll talk about it coming up. Then the chief cells produces a zymogen called pepsinogen, right? Again, zymogen, I highlighted the GEN because these are enzymes and hormones that are produced inactive. They need to be cleaved for them to be activated. So they're usually produced a little bit too long. And then to activate it, you cut apart any excess amino acid, and then that activates them. And then when you no longer need it, you put those amino acids back on. Pepsinogen is one of those zymogens. So pepsinogen is produced by chief cells. Then we have endocrine cells that allows us to release their content into the bloodstream. Things like gastrin are secreted from gastrin-containing cells. Gastrin will go into the bloodstream, go into, remember the bloodstream means that it's going to go into eventually the hepatic portal vein and then back into the stomach. The gastrin then will increase activity of the stomach even more, right? So what we're going to see is before the food has been moved to the small intestine, you're going to have a lot of gastrin be secreted. So now food's only in the stomach. What are we going to do? We're going to make and activate the stomach a lot, right? Once we start secreting food matter, right, that food matter goes from the stomach into the small intestine. We're going to slow everything down in the stomach. So when food starts being moved from one organ to the next, you always slow down the previous organ. So then there's time for that food matter, right, to work its way all the way through into the new one. So the weird thing is this, right? When the food goes into your stomach, you activate the stomach more and more and more. Gastrin's released, so then you can activate the stomach even more. But once we start pushing the food into the next part, the small intestine, the duodenum, then we release somatostatin. Then it inhibits the stomach. So then you don't just force that food into the small intestine quickly, right? Because if you did, each morsel of food is not going to have time to get digested and absorbed. So once food is introduced to the next section, we slow down the previous section. Now, one more thing about the mucus. The mucus here is slightly basic. It's alkaline. That's important because now, right, we have these mucus produced by these cells that are covering the lining of the stomach. And now that mucus is full of base, and it protects the lining of the stomach from the hydrochloric acid we're going to produce by those parietal cells. So then the parietal cells releases the hydrochloric acid into the opening, and then the cells lining the opening now have lots of basic mucus there. That basic mucus protects the stomach from its own hydrochloric acid. Right? Now, you guys have heard of H. pylori. Helicobacter pylori is an infection and it occurs in the stomach, right? I've talked to doctors they talk about like 20, 30 years ago. There's no way they would have thought that a peptic ulcer could be caused by a bacterial infection. But now we have kind of, gotten biopsies and we have seen that H. pylori is present in those peptic ulcers. And what makes H. pylori important is that it uses ammonium as an energy source. So then if you have H. pylori, then what happens? It uses all the ammonia, and now your mucus is no longer basic. It's no longer alkaline. You still have some protection from just the mucus, but without the alkalinity, you can't neutralize the hydrochloric acid anymore. And you don't have the protection from your own acid. And the hydrochloric acid we make starts to digest our own stomach. that happens. Now, one thing about hydrochloric acid activates pepsinogen to pepsin. It cuts the excess amino acids from pepsinogen. It cuts them off. As it cuts it off, it doesn't destroy it. It just cuts it off, right? As it cuts it off, then we activate from pepsinogen to pepsin. And pepsin starts off cutting the amino acids. So I want you to think, right? A protein is long strands of amino acids, and those amino acids then, right, are all kind of bundled together. Because remember, proteins have primary, secondary, and tertiary structure, and they're usually all globuled up, right? Think of a piece of paper that you've just you're about to throw it away. You kind of make it into a ball as you throw it into a wastebasket. Well, that's what the protein should look like, that ball, right? So the ball needs to be unrolled first. And one of the functions of hydrochloric acid is that it unrolls that protein into long chains of amino acids, right? The unrolling means we denature that protein. I know this is talking about chapter 3 material, right? Chapter 2 material as well with primary protein structures. So what we need to do with proteins is when we ingest it, if you're eating a steak, you're eating the skeletal muscle that contains actin and myosin. Those proteins are very, very structured. They're very folded. If they stay folded, the enzymes can't get in there. You need to unfold it. So hydrochloric acid unfolds the proteins. As it unfolds the proteins, it also activates pepsin. And now pepsin can cleave the proteins into smaller bits. So instead of this real long, real folded protein in the stomach, hydrochloric acid unrolls it. So then it's a real long amino acid chain, but it's straight, right? And then pepsin starts to cut it into smaller chunks, right? So instead of an amino acid chain that is 2,000 amino acids long, right? Now pepsin works in there. Now it's in like 5 amino acid chains. That's 400 amino acids in length. Still too long, but at least you started breaking it apart, right? So the weird thing is pepsin is only active in the stomach. The reason why is because pepsin requires an acidic nature. Without hydrochloric acid, it will, right, start turning into zymogen again. Now this picture right here shows us that in order to make hydrochloric acid, you don't make the hydrochloric acid inside the cell. What you do is you pump chlorine out and you pump hydrogen out. Once you pump both of them out, then they combine in the duct of the gastric gland. Then the hydrochloric acid goes up the gastric pits into the opening, that is your stomach. Lining the stomach, basic mucus. So what happens? We never have hydrochloric acid being formed in the cell itself. If it was formed in the cell would die. Instead, they pump out the reactants, the hydrogen and the chlorine. And then by pumping it out, we form hydrochloric acid outside of the cell. We call this a proton pump. Some of you have heard of a PPI, a proton pump inhibitor. Prevacid, Protonix, right? Or Prilosec or Omeprazole. Those are all PPIs. PPIs stop this proton pump and all the proton pumps. Because they stop the proton pumps, you should make less hydrochloric acid because the proton doesn't get pumped out anymore, right? So it's a more efficient way of dealing with GERD. Now, Tegamet will stop antihistamine. It's an antihistamine. So they stop histamine from activating this pump. But there are other ways to activate it. So it's not as strong of a medication as a PPI. PPI stops this pump completely. All right, so the phases of the stomach, and then let's see what time it is, and then we'll stop. right? So what are the phases? And this is interesting stuff. And because I say it's interesting, it's gonna probably be like a written part of a question. So we have these phases: cephalic, gastric, intestinal phases. What do they mean? Now, these are phases that we normally see when somebody is fully functional. You're awake, you're conscious, right? In the cephalic phase, taste, smell, even sometimes sound, right, or thought of food can stimulate the medulla oblongata to then activate the vagus nerve and then activate the stomach. So when you're hungry and you hear your mom and dad or your husband or wife, right? If you hear them start making food right away, right? If you're already hungry, your stomach is going to start to churn. Or if right now you're hungry and I start talking about cheesecake, excuse me, or ribs. Actually, now I'm getting hungry, right? And what happens is you thought of that, and sometimes if you're hungry already, your mouth starts to water already, right? Just the thought of food. That's what we call the cephalic phase. The cephalic phase, cephalic for your head, right, is going to be when other type of sensations besides food entering the stomach, other sensations like taste, smell, thought, right, of food starts to stimulate your parasympathetic nervous system, which is carried by the vagus nerve, right? And then the vagus nerve starts activating the stomach, and then you have increased secretions in the stomach. And what it does is it prepares the stomach to accept the food. It upregulates it. So then by the time the food actually gets into the stomach, it already has a lot of secretions. The stomach muscles are ready to go, ready to contract. So then you don't have to wait, right? The biggest mediator is the vagus nerve. Vagus nerve directly stimulates the parietal and chief cells to produce more hydrochloric acid and pepsinogen, right? It can stimulate the secretion of gastrin and histamine. There's histamine again. Gastrin is carried through back into circulation, and then it goes into the stomach where it further stimulates the stomach even more. So what happens? We have this massive increase in gastric secretions, massive increase in gastric contractions, all due to thought, smell, right? Taste kind of makes sense. When you taste food, you know it's going to come in. But the weird thing is the thought, the smell of food could be enough. The next phase is what happens when the food hits your stomach. Once food hits the stomach, then we'll hit the gastric phase. The gastric phase, right here, right? Food hits the stomach starts to stretch. As the stomach starts to stretch, it activates the vagus nerve. The vagus nerve then increases activity of the stomach. Right? Again, distension stimulates also local reflexes that causes the stomach secretions to increase and the motility to increase. The first two phases increases the activity and secretions of the stomach. So then it's all ready to mix and churn everything and to break down proteins. The last phase is different, intestinal phase. Once the food goes from your stomach, and what I want you to think, this area right here, right? Once food goes right here, there's a sphincter, a valve right here that separates the stomach from the duodenum. That valve will prevent reflux of food in reverse. And what happens is, in order for food matter to go forward, it's going to need to be broken down until it's almost like a soup, a stew, right? This real thick soup then is going to be formed way down here, and it's going to have combinations of all secretions, combination of food matter and carbohydrates, and then your stomach muscles, your 3 layers of smooth muscle, will contract and eject that food from this region called the pyloric region into the duodenum. It is a contraction of the stomach muscles down here that allows you to push that food into the duodenum past that pyloric sphincter, right? Now, Once that food goes into the duodenum, we call that combination of food and liquid, the stew matter, chyme. So chyme is going to be food with all of the juices that we've released, all the hydrochloric acid, all of the pepsinogen or pepsin, all of the enzymes from our mouth, right? All of that combination is called chyme. That chyme gets introduced into the small intestine, and once that chyme gets introduced into that small intestine, that chyme is going to have a lot of acids in it, right? Because of that, the pH of that chyme is very low. It's very acidic. That acidic chyme going into the duodenum then is going to slow everything down. Remember, when we go from one organ to the next, we always slow down the previous organ. So once we introduce food from the stomach, right here, to the small intestine, the duodenum. The start of the small intestine is a duodenum. Once I get introduced into duodenum right here, that food is going to be called chyme, and it's going to be soaked up with a lot of hydrochloric acid, making that chyme very acidic, less than 2 pH. As it goes into the duodenum, it causes the vagus nerve to then decrease the activity of the stomach. Again, why should it make sense? It should make sense, big picture wise, is this. When you eat, you want to break down as completely as possible so then we can absorb as much as possible, because we never knew when our next meal was coming. So what happens? We want that each morsel of food, to get that each morsel of chyme that enters the duodenum, we want it to have time to go through the 20 feet of small intestine so that we can digest and then absorb each morsel of chyme. The faster we unload, the less time for each chunk, each bolus to get worked on and get absorbed. That makes it less efficient. So what we want to do is increase efficiency. Once that first bolus of chyme enters, we slow everything down. So then that chyme, that first bolus, slowly moves through the small intestine, slowly gets worked on by enzymes, and slowly has time to be digested. Perfect, right? That increases a chance of efficient absorption. And that's what happens here. In the intestinal phase, we slow everything down. The vagus nerve slows down its activity, which then slows down gastric secretions. Right? We release secretin and cholecystokinin, which prevents gastric emptying, delays it. So then you're not just pushing that chyme right after one after each other. You slow everything down. We're going to see this again when we go from the small intestine to the large intestine. With the first introduction of waste into the large intestine, we actually slow down small intestine. So then there's more timing. So then we can increase the absorption. Perfect. Right? Now, these highlighted enzymes and hormones right here are really good, right, for multiple choice questions on our next test. So I highlighted it to know which ones I want you guys to know: gastrin, secretin, cholecystokinin, right? Secretin and cholecystokinin produced by the duodenum, and what does it do? Right? We'll talk more about the enzymes and the hormones probably next week. Right now, I'm gonna start wrapping it up. I will be online on Thursday, and I know I have some students that say they can't make it this late. A lot more students say they can, so that's why we're gonna keep our Wednesday meetings from 2:30 to 4:30. And I'm gonna open up a new time frame on Thursday, probably at about 12 o'clock or so. So then, like tomorrow, so then we can go over some anatomy even, and go over and review some of this. So then if you didn't understand it, you might have questions, feel free to ask, okay? You guys have any questions? No? All right, I'm gonna post the lecture on YouTube and then link it. for the next week, all you have for the next week is to, again, review all of the anatomy videos I had posted. And next week, Wednesday, I'll release the lab practical. And once I release it, you'll have like the rest of the semester to get it done. You only have one attempt, but you'll have the rest of the semester in case you, right, run out of data, in case anything happens, right? I want to make sure that you're not going to be handicapped because of the data situation. And you can always email me if you guys have any questions. So what we have right now is basically, you know, for this week, yep, the lab practical will be online as well. So it will be very similar to what we saw on the lecture exam, right? It'll be timed. It'll be also timed and online, just like the lecture exam. And it'll be on the word list that we had. Combination of pictures of models and pictures of cadavers. All right. All right, I'll send a link for tomorrow's, and you guys have a good day, okay?
Thank you. You too.
Bye.
Bye, guys.